Welcome back. Hope everyone enjoyed the break and our SBIA video. We will now move on to our next sessions. This next session by Dr. Cha-Hee Myung will provide an overview of bioequivalence. Dr. Cha-Hee Myung is a pharmacologist at the Division of Bioequivalence 3 in the Office of Bioequivalence within the Office of Generic Drugs here at CDER FDA. In her role, she assesses in vivo bioequivalence studies submitted by pharmaceutical industries through abbreviated new drug applications, or ANDAs, and in vitro bioequivalence studies for a variety of complex dosage forms such as topical cream and gels, nasal sprays, and long-acting injection. Next, we will have a presentation from Dr. Rong Wang. This presentation will provide a broad overview of the scientific and regulatory principles governing bioequivalence with an emphasis on ANDAs. Dr. Rong Wang is currently the Acting Associate Director in the Division of Bioequivalence 1, Office of Bioequivalence within the Office of Generic Drugs. Dr. Wang has worked as a pharmacologist in the Office of Bioequivalence for over 10 years and accrued extensive knowledge and experiences in generic drug and bioequivalence evaluation. So now, please join me in welcoming our next speaker, Dr. Myung.
Thank you for your kind introduction. This is my great pleasure to present the introduction of bioequivalence for generic drug products on behalf of the Division of Bioequivalence 3 in the Office of Generic Drugs today. This presentation reflects the views of the presenter and should not be construed to represent the FDA's views or policies. Learning objectives of current talk are, first, to refresh the concepts of pharmaceutical equivalence and the bioequivalence for a generic drug product. Second, to define a reference standard in abbreviated new drug application and Orange Book. Third, to describe the general considerations in BE studies and product-specific guidance. Fourth, to describe the general considerations in BE studies with pharmacokinetic endpoints. Fifth, to list the drug products and their certain conditions for a waiver of the in vivo BE study requirement. To meet the learning objectives, we will check the concepts of therapeutical equivalence, pharmaceutical equivalence, and bioequivalence in the ANDA submission. In the second section, the fundamental concepts for BE approaches, such as definition of reference listed drug and the reference standard, different types of BE studies recommended in PSG product-specific guidance, overview and general consideration in BE study with the PK endpoint, and the bio-waivers will be covered in the current talk. The mission of the Office of Generic Drugs is ensuring high-quality, affordable generic drugs are available to the American public. Currently, in the United States, 9 out of 10 prescriptions are filled by generic drugs. Compared to its brand name or innovator drug products, average cost of a generic drug is 80 or 85% less. To understand the generic drugs, we need to understand 3 different concepts: pharmaceutical equivalence, bioequivalence, and therapeutic equivalence. Pharmaceutical equivalence is defined as drug products in identical dosage forms and the routes of administration that contain identical amounts of the identical active drug ingredient, that deliver identical amounts of the active ingredients over the identical dosing period, and meet the identical compendia or other applicable standards of identity, strength, quality, and purity. The bioequivalence defined as the absence of a significant difference in the rate and extent to which the active ingredients or active moiety in pharmaceutical equivalents or pharmaceutical alternatives become available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study. The therapeutical equivalence is when 2 pharmaceutically equivalent products are shown to be bioequivalent, The two products are judged to be therapeutically equivalent. Therapeutically equivalent products are expected to have the same safety and efficacy profile when administered under the condition listed in the drug product labeling. Abbreviated New Drug Applications, also known as ANDAs, contain data which are submitted to FDA for the review and the potential approval of generic drug products. Per the Code of Federal Regulation 320.21b, the applicants require submission of evidence that the proposed drug product is bioequivalent to the reference listed drug or information supporting waiver of evidence demonstrating in vivo BE. Per the CFR 320.24a, FDA may require in vivo or in vitro testing or both to measure the bioavailability or establish the bioequivalence of specific drug products. Applicants must conduct the BE testing using the most accurate, sensitive, and reproducible approach available among those set forth in 320.24b. From this section, we will go through the fundamental concepts and the general consideration for BE approaches. Referencing drug products are defined in the guidance named Referencing Approved Drug Product in ANDA Submission Guidance for Industry. This guidance is published in October 2020. It shows that how to identify a reference listed drug RLD and the reference standard RS and the basis of submission in an ANDA submission. The RLD is defined as the previously approved drug product to duplicate. RS is the specific drug product that will be used in conducting any in vivo B testing required to support the approval of ANDA. The basis of submission is a basis for ANDA submission. It should include the name of RLD, dosage form, and strength, and its application number on Form FDA 356H. You can search your RLD or RS products from the Orange Book. It is also known as FDA's Approved Drug Products with Therapeutic Equivalence Evaluations. This is one of the examples. After you search the diclofenac potassium capsule, you can see NDA number 02202. Gypsol capsule is defined. You can see it is identified as RLD or RS for these drug products. At the same time, you can see the TE code. This is the evaluation code. If it is start AA, AN, AO, AP, or AT, that means this drug has no known or suspected BE problems. Or this TE code as shown as here, if it is AB, that means this drug has actual or potential BE problems resolved with adequate in vivo or in vitro evidence supporting BE. Or this TE code start with a B, that means these drug products that FDA at this time considers not to be therapeutically equivalent to other pharmaceutically equivalent drug products. After identifying your RLD and RS product, we need to decide about the BE study design. Multiple things we need to consider. We listed them on this current slide. First, release mechanism of drug substance from the drug products. For example, immediate versus extended release mechanism. Second, intended site of action. For example, systemic versus locally acting drugs. Third, formulation design and composition. For example, tablet versus injection. Fourth, ability to measure the drug availability systemically or at the site of action. For example, plasma concentration versus drug levels in aqueous humor. Fifth, available in vivo and in vitro BE study. For example, dissolution test, in vitro binding test, and in vitro skin permeation test. After considering all these parameters for the BE study design, FDA published the product-specific guidance PSG for generic drug development. We have a website with a searchable function. PSG shows the agency's current scientific thinking on the most sensitive accurate, and reproducible approach to demonstrate the bioequivalence of a generic products to a reference standard products. This PSG is published and revised in a quarterly manner. This PSG may recommend in vivo bioequivalence studies, in vitro BE studies, or a combination of both per CFR 320/24. PSG also may recommend a waiver route. Scientific justifications are needed if the applicants propose the bioequivalence approaches deviated from the PSG's recommendations. More details regarding this PSG development and revision will be covered in the next presentation by Dr. Wang from the Office of Bioequivalence. In the PSG, we recommend the various types of evidence to establish the bioequivalence per CFR 320/24b. In the current presentation, in vivo and in vitro approaches only for the systemically acting drugs, not for the locally acting drugs, will be covered. The first type is the in vivo test in humans measuring active ingredients or active metabolites in appropriate biological fluid as a function of time. It is also known as a comparative pharmacokinetic PK study. Second, in vitro test that has been correlated with and is predictive of human in vivo bioavailability data. Third, in vivo test in humans in which the acute pharmacological effect is measured, also is known as a comparative pharmacodynamic study. Fourth, well-controlled clinical trial, for example, BE study with a comparative clinical endpoint study. Fifth, in vitro test, for example, a comparative dissolution test. And the last, any other method deemed adequate by the agency. These in vivo, in vitro approaches are in descending order of accuracy, sensitivity, and reproducibility. Among multiple in vivo and in vitro approaches in the previous slide, bioequivalence studies with a pharmacokinetic endpoint, also known as a comparative pharmacokinetic study, is mostly and commonly recommended. More details regarding this comparative pharmacokinetic BE study are available in the FDA's guidance, named as Bioequivalence Studies with Pharmacokinetic Endpoints for drugs submitted under an ANDA. This guidance is recently updated in August 2021. It shows how to meet the bioequivalence requirements set forth in FDA's regulations. For most dosage forms that release a drug intended to be systemically available, FDA recommends that the applicants perform a 2-period, 2-sequence, 2-treatment, single-dose crossover study using either healthy subjects or other populations as appropriate. In this design, each subject should receive each treatment, the test or reference product, in a random order. The BE studies generally should be conducted on the highest strength of the drug product, unless safety considerations preclude the use of the dose in studied subjects. As shown in the schematic figure below, after subjects are enrolled in the study, each subject is randomized to one of the 2 sequences. The subjects in sequence 1 receive the test product in the first period and the reference in the second period. However, the subject in sequence 2 receive the reference in the first period and the test in the second period. Between period 1 and 2, a washout period is recommended in this crossover study. There are 2 alternative designs recommended by FDA. First, a single-dose parallel study design. In this parallel design, each subject is randomized to only one treatment. A parallel design is not commonly used in BE studies since it cannot distinguish between inter-subject variability and intra-subject variability because each subject received only one treatment. Under circumstances, parallel design should be used. For example, if the drug has a long half-life, parallel designs may be more appropriate than other designs due to the likelihood of dropouts during the required long washout period. The other alternative design is partial or full replicate design. During 3 or 4 periods, at least one treatment is repeated. A replicate crossover study design, either partial or fully replicate, is appropriate for drugs if the reference drug is a highly variable drug or narrow therapeutic index drug. A replicate design can have the advantage of using fewer subjects compared to a non-replicate design, although each subject in a replicate design study would receive more treatment. For the pharmacokinetic PE studies, 2 single-dose crossover pharmacokinetic studies usually recommended for both immediate and modified release drug products. First one is a PK crossover study under fasting conditions, which is the most sensitive, accurate way to evaluate the formulations. The second crossover study under fat conditions to assure the drug product performs the same way in the presence of high-fat, high-calories meal. Most studies use healthy normal male and female subjects. Sometimes patients are recommended if there are safety issues. The subject numbers in the study should be based on appropriate sample size calculation. Typically more than 12 subject numbers are recommended for general representative. After obtaining the samples from appropriately designed bioequivalence studies, the study samples need to be analyzed using the most accurate, sensitive, and reproducible analytical method. So we published the guidance for this bioanalytical method named Bioanalytical Method Validation Guidance for Industry. This guidance is published in May 2018. It shows how to ensure the bioanalytical quality of bioequivalence data report. In this guidance, we clearly mention that the standard operating protocols, SOPs, related to the validation and analysis should be established before the study starts. The bioanalytical method needs to be validated before the study starts in terms of the selectivity, specificity, and sensitivity, linearity, recovery, accuracy and precision, long and short-term storage stability, and dilution factors. The study samples also need to analyzed with the quality control samples to make sure that all study samples are analyzed under good quality condition. Also, after finish the study sample analysis, we recommend to perform the incurred sample reanalysis, ISR, to verify the reproducibility of this analytical method. Based on the analyzed concentration of active pharmaceutical moiety or active metabolite as a function of time in the BE studies, is statistical analysis for the pharmacokinetic parameters needs to be performed to establish or determine bioequivalence set forth in 21 CFR Part 320. There is a FDA's guidance for the pharmacokinetic and statistical analysis for bioequivalence determination. Its name is Statistical Approaches to Establishing Bioequivalence, which is published in February 2001. This guidance shows the details to provide the equivalence criteria in analyzing in vivo or in vitro bioequivalence studies. The statistical analysis is performed for pharmacokinetic measures such as peak concentration Cmax and the area under the curve AUC based on the two one-side test procedure to determine whether the average values for the pharmacokinetic measures determined after administration of the test and the reference products were comparable. Cmax is defined as the maximum observed concentration during the in vivo bioequivalence study. Compared to other pharmacokinetic parameters, Cmax values are usually have higher variability and highly impacted by the sampling time points in the bioequivalence study design. Area under the drug concentration time curve from the pre-dosing 0 hour until the last time point is a AUCT, which reflects the total exposure of drug to the body up to the last sampling time point. AUC infinite is a theoretical measure of the total exposure of drug to the body from administration until all the drug is eliminated. The area under curves is dependent on the rate of elimination of the drug from the body and the dose administered. To statistically analyze bioequivalence study data, analysis of variance ANOVA with two one-sided test procedure is usually recommended. This approach is called as average bioequivalence, which accesses the variation in the average test and reference difference among individuals. A 90% confidence interval for the ratio of the average of the measures for the test and reference products is calculated. To establish bioequivalence, the calculated confidence intervals for pharmacokinetic parameters such as AUCT, AUC infinite, and Cmax should fall within a bioequivalence limit, usually within 80.00 to 125.00% for the ratio of the product averages. I guess maybe you may wonder why the statistical analysis only for 3 pharmacokinetic parameters is recommended here. The answers are in the definition of bioequivalence. Bioequivalence is defined as the absence of a significant difference in the rate and the extent to which the active ingredients or active moiety in the pharmaceutical equivalence or alternatives becomes available at the site of drug action when administered at the same molar ratio under similar conditions in an appropriately designed study. As you see from the definition of BE, Cmax is related to the rate of absorbed drug, and AUC values are related to the extent of observed drug. Thus, the average bioequivalence analysis for AUCT, AUC infinite, and Cmax is usually recommended. Up to the previous slide, we go through the fundamental concepts and BE approaches, such as a comparative pharmacokinetic BE study and its related bioanalytical methods and statistical approaches. From this slide, let's switch a gear and go through a list of drug products for a waiver of the in vivo bioequivalence study requirement. Per 21 CFR 320.22, a waiver of the in vivo bioequivalence study requirement may be granted for the Biowaiver drug products if certain conditions are met. First, parenteral solution for injection. Second, otic or ophthalmic solution. Both of these solutions products, if they are qualitatively Q1 and quantitatively Q2, the same as the reference listed drug, then these drugs can be waived for their in vivo BE study requirement. Third, oral, topical, nasal solutions, elixir, syrup, tincture, a solution for aerosolization or nebulization, or similar other solubilized forms, they could be granted for the waiver if the formulations has the same active pharmaceutical ingredient in the same concentration and dosage form as the reference listed drug and does not contain any inactive ingredients affecting the drug absorption or availability. The fourth, drug efficacy study implementation that see effective drug products. FDA's administrative process to consider the effectiveness of drug that had been approved only for safety between 1938 and 1962 is called the Drug Efficacy Study Implementation, DESI. Congress amended the Federal Food, Drug, and Cosmetic Act in 1962 to require that new drugs be shown effective as well as safe to obtain FDA approval. The amendment required FDA to evaluate the effectiveness of the drug approved only for safety between 1938 and 1962 when Congress enacted the act requiring new drugs be shown safety prior to marketing. So, FDA and DESI panel reviewed and evaluated the effectiveness of more than 3,400 drugs that had been approved only for safety between 1938 and 1962. FDA's final DESI determination classifies a drug as either effective for one or more of its labeled indications or lacking substantial evidence of effectiveness for one or more of its labeled indications. FDA's finding and a list of DESI drugs had been published in the Federal Register. The drugs in this DESI list are granted for a waiver of in vivo bioequivalence study, but adequate dissolution data is still required. The fifth is Biopharmaceutics Classification System, BCS-based biowaivers. We have a related guidance, M9 BCS-based biowaivers. It is published in May 2021. BCS is a scientific approach based on the aqueous solubility and intestinal permeability characteristics of the drug substances. Based on this scientific approach, we waive two different classes, BCS Class 1 with high solubility and high permeability, and Class 3 with high solubility and low permeability. More details regarding about these BCS-based biowaiver drugs will be covered in the next presentations by Dr. Rong Wang from the Office of Bioequivalence, as well as by Dr. Yi Zhang from the Office of Research and Standards. The sixth, non-bio study strengths of solid dosage forms for immediate release products could be waived under 21 CFR 320-22D2. It needs three specific conditions. First, adequate in vivo bioequivalence study on the biostrengths. Second, adequate in vitro dissolution test across all strengths. And third, the proportional similarity of the formulations across all strengths. In the case of non-bio study strengths of solid dosage forms for modified release products, should be demonstrated to be bioequivalence under 21 CFR 320-24B6. It also requires some specific conditions too. First, adequate in vivo bioequivalence study on the biostrengths. Second, adequate in vitro dissolution test across all strengths in at least 3 dissolution media, for example, a pH of 1.2, 4.5, and 6.8. And third, qualitatively sameness, quantitatively similarity with justification is required. That means proportional as well as non-proportionally formulated strengths could be justified. And the last, the same drug release mechanism across all strengths are required. In summary, a generic drug must be pharmaceutically equivalent and bioequivalent and thus therapeutically equivalent to the brand name drug product. Related regulations and resources for development of generic drug products, such as the product-specific guidance, Orange Book, guidance documentations for industry, are available to the public. Comparative pharmacokinetic studies, for example, single-dose crossover bioequivalence studies, are mostly used for bioequivalence demonstration of systemically absorbed generic drug products. The applicants can request waivers of in vivo bioequivalence studies if their biowaiver drug products meet FDA's criteria. Here are 2 poll questions. First, Which of the following study design is recommended for bioequivalent studies of a drug substance with a very long half-life? A. Single-dose two-way crossover study design. B. Single-dose parallel study design. C. Single-dose replicated study design. D. None of the above. The answer is C. Single-dose replicated study design. This is recommended for the drug substance with a very long half-life. Poll question 2. Which of the following drug product cannot be considered for a bio-waiver? Waiver of in vivo testing. A. Parenteral solution for injection, which is quantitatively the same as reference listed drug. B. Auric solution, which is quantitatively the same as RLD. C. Drug efficacy study implementation that's effective drug products. D. Non-bio study strengths of solid dosage form with inadequate bioequivalent studies on the bio strengths. The answer is D. Non-bio study strengths of solid dosage forms with inadequate bioequivalent studies on the bio strengths. It required adequate bioequivalent studies on the bio strengths. Here is the resources what I cited during my presentation. All guidances are available to the public. With that, I would like to thank you for your attention and all helps from the Office of Bioequivalence here. I am happy to answer any questions you may have in the panel section. Thank you.
Good morning, everyone. It is an honor to be here today. My name is Rong Wang. I'm representing the Office of Bioequivalence in the Office of Generic Drugs at FDA. The topic of my presentation today is bioequivalence studies for generic drug development. The disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. In today's presentation, I will briefly talk about the role of bioequivalence in generic drug products, then give a quick overview on regulations that govern the bioequivalence determination of a generic drug product. Then, I will talk about some of the tools that the agency provides to help industry develop generic drug products. One of the tools is FDA general guidances that are relevant to bioequivalence studies. I will highlight some examples of the general guidances in the presentation. In addition to the general guidances, another useful tool FDA provides is Product-Specific Guidances, or PSG. I will touch upon considerations in developing a PSG and highlight the key elements that are usually included in a PSG. Then towards the end of the presentation, I will provide a high-level overview on bioequivalence information that should be submitted in an abbreviated new drug application, or ANDA, and common issues for BE studies. Lastly, I will summarize some key takeaways from today's presentation. As you have learned from the previous talk, 21 CFR 314(3)b laid out the critical foundation of therapeutic equivalence. So, generally, in order to be approved, a generic drug must be pharmaceutically equivalent and bioequivalent to the reference listed drug product, and therefore therapeutically equivalent to the RLD. I will not go through the definitions of pharmaceutical equivalence and bioequivalence as they were covered in the previous talk. I just want to highlight here that a therapeutically equivalent generic drug product is expected to have the same clinical effect and safety profiles when administered to patients under the conditions of use specified in the labeling. There is a number of regulations that govern bioequivalence studies. I'm not going to go through all of them, but here I just highlight a few of them. Some regulations like 314-94A7 and 320-21B give the agency the authority to ask for bioequivalence documentation in support of an ANDA or information to support bioequivalence or a waiver of in vivo bioequivalence studies. CFR 320-22 contains requirements regarding criteria for a waiver of in vivo bioequivalence studies. CFR 320-23B contains requirements of bioequivalence stating that two drug products will be considered bioequivalent if rate and extent of absorption do not show a significant difference when administered at the same molar dose of the active moiety under similar experimental conditions. It also states that bioequivalence may be demonstrated by scientifically valid methods for drug products not intended to be absorbed into the bloodstream. CFR 320-24A has two parts. The first part of this reg says FDA may require in vivo or in vitro testing, or both, to measure the bioavailability of a drug product or establish the bioequivalence of specific drug products. It also emphasizes that applicants should conduct bioavailability and bioequivalent testing using the most accurate, sensitive, and reproducible approach available. In this regard, there is a number of different types of studies that are out there to support bioequivalence of a generic drug product. So 21 CFR 320-24B outlines the general approaches for demonstrating bioequivalence as summarized here in this slide. The methods for establishing BE can range from an in vivo pharmacokinetic study where you are measuring active ingredient or active metabolite in a biological fluid such as blood to an in vivo pharmacodynamic study where you measure the pharmacological effect of a drug product or a clinical endpoint study. Sometimes you may need in vitro tests to establish BE under appropriate circumstances. Again, to highlight the point, different approaches may apply to different drug products for demonstrating bioequivalence. That is also in line with CFR 320-24A, which says the most accurate, reproducible, and sensitive approach must be used to demonstrate bioequivalence. Next, I'm going to talk about some FDA general guidances that the agency provides to help industry develop generic drug products. The first guidance I'm going to talk about is the guidance referencing approved drug products in ANDA submissions. This guidance was published in October 2020. It provides information to potential generic applicants on how to identify a reference-listed drug or RLD, a reference standard or RS, and the basis of submission in an ANDA. Dr. Miang has provided in her talk the definitions of RLD and RS and where to identify and verify RLD and RS. Just to briefly highlight the point, RLD is usually the basis of submission for an ANDA, and RS is the drug product that applicants use as a comparator in conducting an in vivo bioequivalence study. RS could be the same as the RLD. However, when RLD is discontinued from the market, the agency may designate a new RS, which is different from RLD. If an applicant has a question about identification of RLD or RS, the applicant may submit control correspondences to seek input from the agency. The next guidance I'm going to talk about is bioequivalence studies with PK endpoints for drugs submitted under an ANDA. This is probably, I would say, one of the most frequently referenced general guidance for generic drug development. This guidance has been updated for several times by the agency, and the most recent version was published in August 2021. This guidance describes general recommendations in study design and data handling of PKBE studies. The guidance laid out the criteria for waiver of in vivo BE requirements for additional strengths of immediate release drug products, and the criteria for demonstrating bioequivalence for additional strengths of modified release drug products. It also includes special considerations for establishing bioequivalence for some special dosage forms, for example, chewable tablets, orally disintegrating tablets, sublingual and transdermal drug products. The guidance provides instructions on how these drug products of special dosage forms should be administered in the PKBE study. The guidance also addressed some other special topics. For example, in what situations metabolites need to be measured in the PKBE study, and what is the role of metabolites in establishing BE. When a drug product is racemic mixtures of enantiomers, under what circumstances individual enantiomers should be measured. How to determine bioequivalence for endogenous compounds. How to treat first time point Cmax in a PKBE study, and alcohol dose dumping of certain modified release drug products. Bioanalytical method validation general guidance is also another frequently referenced guidance. The most current version of this guidance was published in May 2018. It provides detailed recommendations on what bioanalytical parameters should be validated for an in vivo BE study. It also recommends acceptance criteria for each parameter to be validated. The guidance laid out what documentation for method validation and bioanalytical reports are necessary for an ANDA submission. This slide here lists examples of the validation parameters. I'm not going to go through it one by one, just to point out here that the guidance includes not only the recommendations for pre-study method validation, but also for parameters for in-study conduct, such as incur sample analysis and the study sample repeat analysis. The guidance also touched upon some special considerations, for example, analysis of endogenous compounds. Next is M9 Biopharmaceutics Classification System-based Biowaivers. This guidance provides recommendations to support the BCS-based Biowaiver. To highlight the point here, the guidance clarified that only immediate release solid oral dosage forms and suspensions are considered eligible for a BCS waiver. Modified release drug products, narrow therapeutic range drugs, dosage forms intended for absorption in the oral cavity, such as sublingual or buccal tablets, are not qualified for BCS waiver B approach. The table on the right side of the slide summarizes the criteria for BCS-1 waiver and BCS-3 waiver respectively. A BCS-1 waiver is applicable when the formulation of the test product does not contain excipients in amounts that affect absorption, the drug substance is highly soluble and highly permeable, and the drug product is rapidly dissolving in multi-pH media. For a BCS-3 waiver, the applicant needs to demonstrate its test formulation is qualitatively the same and quantitatively very similar to that of the RLD product. The drug substance is highly soluble and the drug product is very rapidly dissolving in multi-pH media. These slides listed some additional general guidances that are relevant to generic drug development. Please note that this is not an exhaustive list. You could go to Guidance Document search webpage to search any relevant guidance documents. I have provided the link of the webpage here. As I mentioned earlier, there is a number of different types of studies that can be used to establish bioequivalence. So how will we know what is the most appropriate method the agency thinks for a specific drug product? To assist the generic drug industry in identifying the most appropriate BE methods, the agency published the product-specific guidances, or PSGs, to describe the agency's current thinking on the evidence needed to demonstrate that a generic drug is therapeutically equivalent to the corresponding RLD product. As you may know, Office of Generic Drugs, or OGD, leads PSG development in collaboration with multiple disciplines and offices within the FDA. Generally, the PSGs are published quarterly. As of July 2022, the agency has published over 2,000 PSGs. The agency has published a PSG database arranged by active ingredient. I listed the link for the database search webpage here. In addition, the agency also maintains a webpage which provides information related to upcoming new and revised PSGs for complex generic drug products. This webpage will be updated each time FDA issues a new quarterly PSG batch. This will help generic industry to plan for their development of complex generic drug products. There is a number of different considerations when we think about what the appropriate bioequivalence approach is going to be for a specific drug product. This slide lists some thoughts to be considered when developing BE recommendations for a systemically available drug product. In general, PK/BE studies are recommended, but for some drug products like inhalational drug products that are systemically available but locally acting, both PK and clinical endpoint studies or PD studies may be needed to establish BE. The route of administration can also affect what studies are needed in addition to regular PK/BE studies. For example, if RLD labeling indicates the drug can be administered through NG tube, in vitro NG tube studies may be needed to ensure the performance of test product through NG tube is comparable to the RLD. If the capsules are recommended to be sprinkled in applesauce for administration, a sprinkle PK/BE study will be considered necessary. Formulation design also plays a role in BE recommendations. For example, for abuse deterrent opioids, abuse deterrence testing is recommended to evaluate if the test formulation has comparable abuse deterrence properties as compared to RLD. For liposome injectables, in addition to in vivo PK study, the applicant should also demonstrate equivalent liposome characteristics, including liposome composition, state of encapsulated drug, liposome size distribution, etc., between test and RLD products. Other considerations are properties of drug substance. For example, for a drug product of endogenous compound, baseline is usually measured in a PK/BE study, and the bioequivalence is usually based on the 90% confidence interval calculated from baseline corrected data. For a highly variable drug product, a replicated study design is usually recommended, and bioequivalence may be established through a reference scaled BE approach. Mechanism of drug delivery can be important to BE recommendations as well. For example, dry powder inhalers are considered drug-device combination products. The drug is delivered through an inhaler, so among various BE studies, in vitro tests such as aerodynamic particle size distribution and a single actuation content are recommended to characterize the drug release performance from the device. These slides highlight some considerations we should think when developing a PSG for a locally acting drug product. For locally acting drug products, an in vivo PKBE study may be recommended if systemic bioavailability is measurable and considered as a surrogate to the bioavailability at the site of action. However, a lot of locally acting drug products may not have measurable systemic absorption. A clinical endpoint study or PD study or some in vitro tests may be used to establish bioequivalence depending on the mechanism of action or site of action and availability and feasibility of the testing. For example, for topical corticosteroids, vasoconstrictor assay is recommended for demonstrating bioequivalence. This approach is based on the property of corticosteroids to produce blanching of the skin. This property presumably relates to the amount of drug entering the skin and thus becomes a possible basis for the comparison of drug delivery from two formulations. Cevalomide carbonate tablets is a phosphate binder. It binds to the phosphate in food before they can be absorbed. So, based on the mechanism of action, the in vitro binding studies is recommended to compare the phosphate binding capacity of test and RLD products. As you may know, a clinical endpoint study has been typically recommended to establish BE for a topical dermatological drug product. However, the agency has recommended in vitro characterization-based BE approaches as an alternative to more and more topical dermatological drug products. This alternative BE approach is based on the knowledge of the formulation and drug release mechanism and understanding of correlation between drug permeation in an IVPT study and the local bioavailability of the drug. In cases where IVPT is not feasible, for example, for some topical foam products, in vitro characterization-based BE approach may not be considered appropriate. The next two slides will highlight some key elements of PKBE study recommendations in the PSG. In general, a PSG with PKBE study recommendations will include study design, study subjects, and study type. Then, what analyte should be measured? Should parent drug or metabolite or both be measured? What type of biologic matrix should be used to measure the analyte? PSG also recommends what analyte would be considered pivotal to BE determination and which analyte, if applicable, should be considered supportive. For a drug product with multiple strengths, the PSG also includes waiver criteria for additional strengths, if applicable. Sometimes the PSG may include additional comments for some special considerations. For example, for endogenous compounds, there might be comments regarding baseline measurements, diet restriction, and baseline correction. For some modified release drug products, the PSG may include detailed instructions on measurement of partial AUC. To this point, we have covered some of the basic regulatory and scientific concepts behind demonstrating bioequivalence, and we highlight PSG databases and some general guidances which provide help to generic drug development. To close this presentation, I would like to take a few minutes to provide a quick overview on BE information that is needed in ANDA submissions and common deficiencies observed by our assessors in Office of Bioequivalence. We hope these could help you submit complete and high-quality bioequivalence submissions in your ANDA. This slide listed two guidances regarding ANDA submissions. One is ANDA submissions content and format. The other one is good ANDA submission practices. Both guidances are intended to assist applicants in preparing ANDAs for submission to FDA, which provide detailed information that should be provided in each section of the common technical document format and highlights common recurring deficiencies that may lead to a delay in the approval of an ANDA. Typically, an ANDA consists of 4 modules all of which contain BE-relevant information. In Module 1, the basis of submission tells you what drug product the applicant is intended to duplicate. Module 2 includes summary tables of BE data. The component and the composition table of the test formulation is usually included in Module 3. Module 5 includes study reports and the protocols of BE studies. One critical point to highlight here is that FDA regulations require that an ANDA applicant to submit data from all BE studies the applicant conducts on the drug product formulation submitted for approval. Either a complete study report or a summary report should be submitted for each individual BE study conducted on the same formulation as the exhibit batch. This includes studies that did not show the product met BE criteria. We believe that data from any additional BE studies may be important in our determination of whether a product is bioequivalent to the RLD. The agency has a summary table webpage. Summary tables for different study types are available on this webpage. For example, summary tables for in vivo PK/BE studies, BCS-based biowaivers, in vitro binding studies, etc. In the next few slides, I'll give a brief overview on bioequivalence information that is needed for an ANDA. For both in vivo and in vitro bioequivalence studies, applicants should submit pre-study method validation report, analytical report for BE studies. The reports are expected to be complete and comprehensive. Applicants should submit analytical raw data from the study runs of all subjects, including accepted runs and rejected runs. Applicants should also submit serially selected chromatograms for at least 20% of the study subjects, if applicable. It is also important for the applicant to submit clear and well-written SOPs used in the application. Please note that SOPs should be effective at the time of study conduct. For study site information, the detailed street address should be provided in the submission for both clinical and analytical sites where BE studies were conducted. For in vivo BE studies, the applicant should submit study protocols, study reports, which include study information, product information, study design, demographic profiles of subjects, dropout information, study adverse events, protocol deviation, and statistical results. The applicant should submit study datasets in appropriate CDISC format and case report forms of all subjects. For in vitro BE studies such as IVRT, IVPT, and QCRT, in addition to a complete pivotal BE study report, the applicant should provide a comprehensive method development report and a complete method validation report to support their selected method. In the next two slides, I'd like to relay to you some common issues that have been noted by your bioequivalence assessors that they would like to share with you. For example, for both in vivo and in vitro BE studies, during assessment of analytical data, the following deficiencies are commonly noted: inadequate dilution integrity data, inadequate stability data, incomplete or missing numerical raw data, incomplete or missing SOPs, missing rejected and/or accepted chromatograms, inconsistencies between summary tables and study reports. For statistical analysis, no pre-specified statistical plan, incorrect model was used, or missing justification for the method used for statistical analysis. For in vitro BE studies, it is often noted that the ANDA submissions does not have study details such as study procedures, apparatus information, number of replicates, and so on, or the applicant did not provide study site information, including name and/or street address. discrepancy within submission, for example, information not consistent between summary tables and the study report, inadequate rationale for selecting method parameters in method development report, or inadequate method validation. I'd like to conclude my presentation by highlighting some take-home messages. Generic drug products approved via an ANDA submission are considered therapeutically equivalent to the ILD based on the demonstration of bioequivalence and pharmaceutical equivalence. Today's presentation was focused on the bioequivalence studies for generic drug development. I touched upon some tools the agency provides to help industry to develop generic drugs. There are general guidances and product-specific guidances, or PSGs. These tools reflect FDA's current thinking for generic product development. BE approaches utilizing in vivo or in vitro methods, or both, may be recommended, depending on the properties of the drug product. For example, site of action, route of delivery, dosage form, formulation design, etc. Also, a detailed and well-organized ANDA submission can reduce the number of deficiencies and review cycles. I'd like to end my presentation with my sincere thanks to my managers and colleagues in Office of Bioequivalence at OGD for their helpful support and guidance on my slides preparation, and SBIA Planning Committee for this great presentation opportunity. Next, let's move on to challenging questions. Challenging question 1. Which of the following should be considered when making B recommendations during PSG development? A. Route of administration. B. Formulation design. C. Site of action. D. Feasibility to measure systemic bioavailability. E. All of the above. The correct answer is E, all of the above. Challenging question 2. Which of the following BE-related information should be included in an ANDA submission? A. Basis of submission. B. Composition and component table of the test formulation. C. Summary tables of B data. D. Reports of failed B studies on the same formulation as the exhibit batch. E. All of the above. The correct answer is E, all of the above. Thank you for your attention.
Thank you again to Drs. Myung and Wang. They will be back to join our Q&A panel just a bit later today. Now it is my pleasure to introduce our next group of presentations and speakers. Next, we will hear from Commander Ji Zhang, whose presentation will discuss essential elements of the Biopharmaceutics Classification System, or BCS, 3 Biowaiver requests to be assessed for generic drug development and approval. Commander Zhang is the Senior Advisor for Oral Drug Products in the Office of Generic Drugs as a subject matter expert in developing and establishing optimal and rigorous BE standards and approaches for various drug dosage forms to promote generic drug development and approval. She has led multiple teams in the Office of Research and Standards. The next presentation that follows will be by Dr. Donna Volpe, which will provide information on how to conduct solubility, permeability, and dissolution according to the BCS guidance. Dr. Volpe is the principal investigator in the Office of Clinical Pharmacology's Division of Applied Regulatory Science. She has experience in vitro assays for drug permeability, metabolism, and transport. She has also been involved in the revision of FDA's BCS Biowaiver and in vitro drug-drug interaction guidances for industry. And finally, we will have a presentation by Dr. Haritha Mandula, which will provide a broad overview of the Biowaiver aspects in NDA and AND post-approval applications. Dr. Mandula is a senior pharmaceutical quality assessor in the Division of Biopharmaceutics within O&D OPQ in the FDA's Center for Drug Evaluation and Research. She has experience supporting the review of biopharmaceutical aspects of new and generic drug product review submissions across a wide range of therapeutic areas. So please join me in welcoming our next speaker, Commander Yi Zhang.
The title for my today's presentation is Essential Elements of BCS3-based Biowaiver Request. Disclaimer: this presentation reflects the views of the author and should not be construed to represent FDA's views and policies. Our three learning objectives based on my today's talk. First, to discuss essential elements of biopharmaceutics classification system, also called BCS, 3-based Biowaiver as an alternative bioequivalence approach. Second, to share research results on potential BCS3 drug products. Third, to investigate formulation impact on BCS3-based Biowaiver. In May of 2021, FDA published a guidance for industry, ICH-M9, on biopharmaceutics classification system-based biowaivers. The link to this guidance is provided on this slide for your convenience. BCS is a scientific framework to classify drug substances based upon both aqueous solubility and intestinal permeability. There are four classes of BCS drug substances: 1, 2, 3, and 4, based upon solubility and permeability. As shown in this table, it has clearly delimited the class boundaries. The BCS-based biowaiver is only applicable to immediate release, solid orally administered dosage forms, or suspensions designed to deliver drug to the systemic circulation. Fixed dose combination, also called FDC product, are eligible for BCS-based biowaiver when all drug substances contained in FDC meet the criteria. Drug products having a narrow therapeutic index or with buccal sublingual absorption are not eligible. Furthermore, the BCS-based biowaiver approach is applicable only when the mode of administration includes water. If the oral IR product contains a drug substance with high solubility, either BCS class 1 or 3, the rate and extent of drug absorption will not depend on drug dissolution and/or gastrointestinal transit time. Therefore, it may not be necessary to demonstrate in vivo bioavailability or bioequivalence as long as the excipients of the drug product do not significantly affect the absorption of the active pharmaceutical ingredients. Overall, BCS biowaiver can surely serve as a beneficial alternative bioequivalence approach or a surrogate for in vivo bioequivalence to the IR oral products by reducing the study costs and unnecessary human drug exposure, eventually expedite the process to promote generic drug development and approval. FDA publishes product-specific guidance, also known as PSG, quarterly for individual drug product to provide bioequivalence recommendation. At the current FDA PSG website, there are more than 30 published PSGs which contain BCS Class 1 waiver option, and 2 PSGs contain BCS Class 3 waiver option, including pentosan polysulfate sodium capsule and hydroxychloroquine sulfate tablets. Here is the agency's general practice for PSGs. FDA only recommends BCS-based Biowaiver as one of bioequivalence options when the drug has been classified by the agency. When the BCS Biowaiver option is not included in the current published PSG to a specific drug, that only means it has not been classified. However, the applicant still can submit its BCS-based Biowaiver request to the agency for further assessment as long as it submitted sufficient supportive data. As we all know, many IR oral drugs having high solubilities which are potentially eligible for BCS Biowaiver, either BCS Class 1 based or BCS Class 3 based. The agency strongly recommend the applicant to show their due diligence. In order to be eligible for BCS Class 3 based Biowaiver, what are the criteria? As per the ICH M9 guidance, the following should be demonstrated. First, highly soluble. The volume required to dissolve drug substance, also referred as BCS volume, should be less than or equal to 250 ml aqueous media over the pH range from 1.2 to 6.8. Second, very rapidly dissolving across multiple pH. As the innovators of many older reference products haven't submitted the dissolution testing data in multiple pH media, the potential applicants need to first determine if the ROD product is rapidly dissolving across the pH range, and then demonstrate the very rapidly dissolving across multiple range for test product. Last, qualitatively Q1 the same and quantitatively Q2 similar to the reference product except for film coating and capsule shell exhibit. In the next few slides, I will elaborate a little bit more detail to each of the essential elements for BCS3 based Biowaiver requests. First, for high solubility. As I mentioned earlier, a drug substance can be classified as a highly soluble when the highest single therapeutic dose is soluble in no more than 250 ml of aqueous media over the pH range from 1.2 to 6.8. In the cases where the highest single therapeutic dose does not meet this criterion, but the highest strength of the reference product is soluble under the required conditions, then additional data should be submitted to justify the BCS-based Biowaiver approach, demonstrating of dose proportional PK, that is, rate and extent indexed by the AUC and Cmax over dose range that include the highest single therapeutic dose. The BCS volume is calculated by HSTD divided by solubility. And again, the BCS volume cannot be more than 250 ml. Again, I mentioned earlier, BCS waiver only eligible when water is included for the mode of administration. Third, a sufficient number of different pH values should be selected based on ionization of the drug substance. For example, pH at pKa, pH at pKa minus plus 1, pH at 1.2, and 6.8. Please keep in mind that the selected pH should stay within the physiological range. And the applicant should demonstrate that solubility is maintained over relevant time frames to accommodate the expected duration of absorption. In addition, adequate stability should be demonstrated as well. In case where the drug substance is not stable with greater than 10% degradation over the extent of the solubility assessment, solubility cannot be adequately determined, therefore the drug substance cannot be classified. And 37 plus minus 1 Celsius degree is the solubility study temperature should be. The second essential element for BCS3 based Biowaiver is very rapid dissolution. So first, USP apparatus, one basket, or two paddle is commonly used for dissolution testing. The agitation speed are 100 RPM for basket and 50 RPM for paddle. When high variability or coning is observed with a paddle speed of 50 RPM for both reference and the test products, the applicant may use a basket speed of 100 RPM. In addition, alternative methods such as the use of sinkers or other appropriately justified approaches may be considered to overcome issues such as coning. Again, the temperature for dissolution testing is the same as the solubility study, 37 plus minus 1 Celsius degree. The volume of dissolution media is 900 ml or less. It is recommended to use the volume selected for the quality control QC test. For the dissolution media include multi-pH 1.2, 4.5, 6.8 buffers. Organic solvents are not acceptable, neither for the surfactants. The dissolution spec is not less than 85% within 15 minutes in all three buffers. For FDC product, if it only contains BCS class 3 drug substances, same dissolution spec apply. However, if the FDC product contains both BCS class 1 and 3 drug substances, each of them should meet the dissolution spec respectively. For example, for BCS class 1, not less than 85% within 30 minutes. For BCS class 3, not less than 85% within 15 minutes. The last essential element of BCS class 3 based Biowaiver is the formulation similarity assessment. The test product should be Q1 the same and Q2 similar to the reference product. Excipients which may affect absorption should stay within plus minus 10% of the amount of each corresponding excipient in the reference product. So does the total additive difference, which are caused by changes in all excipients as shown in this table, which is cited from ICH M9 guidance. In the meanwhile, please keep in mind that differences in colorants and flavoring may be permitted when they only constitute very small amounts of the formulation. This is a decision tree from ICH M9 guidance on formulation similarity assessment. BCS Class 3 drug substances are commonly considered to be more susceptible to the effects of excipients. If there are existing excipient differences in the test formulation, in order to be eligible for BCS Class 3 based Biowaiver, the applicant should justify why the proposed excipient differences will not affect the absorption profile of the drug substance, that is, rate and extent of absorption, by using a mechanistic and risk-based approach. Impact of excipients on solubility, gastrointestinal mobility, transit time, and intestinal permeability, including transporter mechanisms, should be considered, as well as the amount of excipient used, the mechanism by which the excipient may affect absorption, and absorption properties of the drug substance, that is, rate, extent, and mechanism of absorption. For example, some excipients may affect absorption. The first one comes to my mind is sugar alcohols, including mannitol and sorbitol. Another one is surfactants such as sodium lauryl phosphate. There are potential challenges in applying BCS Class 3 based Biowaiver. The agency has initiated multiple research projects to address the challenges in order to promote the application of BCS Class 3 based Biowaiver for generic drug development and approval. Currently, two key limiting factors are subjects of research. Challenge 1: to meet the criteria for very rapid dissolution. As I mentioned earlier, the dissolution testing data in multiple pH media for many old RODs are not available. Therefore, we have started a collaborative research project to generate solubility data and multiple pH media dissolution testing data for oral IR drug products with BCS Class 3 potential. Challenge 2: to meet the criteria for formulation similarities. Research Project A: expanding BCS Class 3 biowaivers for generic drugs to non-Q1, Q2 formulations. Research Project B: excipients impact on bioavailability of BCS Class 3 drugs. Research Project C: assessment on the formulation similarity of approved generic drug products with BCS Class 3 potential. Research Project D: physiologically based pharmacokinetic, also known as PBPK, absorption modeling as an alternative bioequivalence approach to support BCS Class 3 biowaiver. Research Project E: effect of excipient transporter interactions on BCS Class 3 drugs. Research Project F: effect of excipients on the oral absorption of fexofenadine in humans. I will provide research outcome brief summary for all research projects except The project 2F, as it just started, currently still at the stage of method validation. For research project 1A on multiple pH dissolution study of potential BCAS class 3 drug products, we have evaluated the dissolution profiles under multiple pH media of a total 14 drug products of 5 drug substances, including atenolol, acyclovir, metformin, hydroxychloroquine, and tenofovir. Among each drug products, at least 2 or more approved generic drug products were studied. For tablets of atenolol, acyclovir, and metformin hydrochloride, each product line contains 2 or more strengths. For tablets of hydroxychloroquine sulfate and tenofovir disoproxyl fumarate, we have conducted multiple pH dissolution studies for both reference-listed and generic drug products. Each product line only has a single strength. In this table, we summarize the drugs we studied in this project, formulation similarity assessment between generic and reference products, as well as their dissolution performance. The results of this multiple pH dissolution test on BCAS class 3 drug products are the following. First, the majority of the dissolution studies demonstrated very rapidly dissolving in all dissolution media as well as meeting BCAS class 3 Biowaiver dissolution criteria. Second, the small percentage of dissolution studies that did not meet the BCAS class 3 Biowaiver dissolution criteria may be due to the dose strength or minor differences in manufacturing procedure, such as the tablet hardness. Lastly, our data show that different dissolution rates, either very rapidly or rapidly dissolving, did not appear to impact the in vivo performance of the approved generic drug products. For research project 2A, which explored the impact of excipients on non-Q1-Q2 formulations, effects of common excipients on both dissolution and permeation of BCAS class 3 drug products were evaluated by in vitro dissolution absorption system, abbreviated as IDAS. IDAS can simultaneously measure both dissolution and permeation of finished drug products in either tablet or capsule dosage forms. 4 pairs of reference-listed and generic drug products that had previously demonstrated bioequivalence in clinical studies were investigated by IDAS, including acyclovir, cimetidine, ranitidine, and atenolol. In this research project, scientists use a novel in vitro product characterization tool to assess the impact of excipients on the dissolution and the permeation of BCAS class 3 model drugs. In addition, they investigated the potential to expand BCAS class 3 biowaivers for non-Q1-Q2 generic drugs by changing the amount of excipients. This is a research outcome summary on the current stage of Project 2A. 7 excipients containing hydroxypropyl methylcellulose with 2 viscosities, microcrystalline cellulose, cross-carbamylose, sodium, TAC, mannitol, and silicon dioxide had no effects on the permeation of any model drugs. Another 6 excipients containing providin K30, magnesium stearate, lactose, calcium phosphate, pre-gelatinized starch, and PAC 400 had some effect, either increasing or decreasing the permeation on only one or two of the model drugs. One excipient, sorbitol, was inconsistent because it affected the permeation of all model drugs, but with mixed effects of either decreasing or increasing permeation. Sorbitol is known to reduce drug absorption in vivo by decreasing intestinal transit time, while it is not known if it affects the intrinsic permeability of drugs. The only excipient that produced dose-dependent increasing effects on the permeation of all model drugs was sodium lauryl sulfate, also known as SLS, which is not surprising because it is a permeation enhancer. For research project 2B, which explores the impact of excipients on the bioavailability of BCS Class 3 drug products. This study used cimetidine and acyclovir as model BCS Class 3 drugs in 4-week crossover bioequivalence studies in healthy subjects. The study was to evaluate the impact of larger than conventional amounts of 14 commonly used excipients on the absorption of BCS Class 3 drugs. Overall, 12 common excipients were found in larger amounts who did not impact BCS Class 3 drug absorption in vivo. The excipients don't need to be Q1 the same nor Q2 similar to the reference product, but rather simply be not more than the certain quantities used in the study. The Q1 sameness of Q2 similarity of task products to the reference product with regard to excipients of hydroxypropyl methylcellulose and microcrystalline cellulose same instrumental. For research project 2C, formulation assessment of approved generic drug products with BCS Class 3 potential. First, we retrospectively collected formulation data in approved generic drug products that successfully demonstrate in vivo bioequivalence. Then we compared compositions between generic and reference drug products. Last, we explored the impact of excipient changes on in vivo bioequivalence outcome. This table summarized all drug substances were assessed in Research Project 2C. A total of 134 approved ANDAs for tablet and capsule products were examined for 13 potential BCS Class 3 drug substances, which having low or moderate permeability ranging from 10 to 83%. Among them, atenolol, colchicine, ranitidine, and tenofovir are substrates of P-glycoprotein. In addition, tenofovir is also a substrate of breast cancer-resistant protein. Both are efflux transporter. By comparing the formulations of this approved generic product, we divided them into 4 groups: Q1, Q2 same, Q1 same, Q2 similar, Q1 same, Q2 different, and Q1 different. If you look at the pie chart on the left, among 134 approved generic drugs, 5% of studied generic formulations were Q1, Q2 the same to the corresponding ROD. 20% were Q1 the same and Q2 similar to the corresponding ROD. By combination of 2 categories, a total of 25% meet formulation similarity criteria set in ICH M9 guidance. 18% of studied generic formulations in approved N does, were Q1 the same and Q2 different to the corresponding ROD. 57% were Q1 different. By adding the two categories, will give a total 75%, which does not meet formulation similarity criteria set in ICH M9 guidance. Therefore, the observed Q1, Q2 formulation differences in excipients of approved generic drug products appear not impact their in vivo bioequivalence performance. Therefore, the formulation similarity assessment criteria for BCS Class 3 based Biowaiver may be warranted for further exploration. This table shows range of top 20 common excipients in potential BCS Class 3 drug products with passing bioequivalence studies. A total of 42 excipients have been assessed in this project. Changes in excipients don't seem to impact the absorption of potential BCS Class 3 drug products. The data on common excipients are just an observation based upon the approved generic drug products. At least the ranges of changes in these excipients don't seem impact the in vivo performance of drug products. However, it doesn't mean that these excipients can be used in all BCS3 products. For Research Project 2D, PBPK absorption modeling is used to explore the impact of gastric pH on absorption of weak base drugs such as saxagliptin. In addition, the virtual bioequivalence simulations were conducted to establish dissolution safe B space for oseltamivir phosphate and its metabolite, oseltamivir carboxylate, in both adult and pediatric populations. For Research Project 2E, the effect of excipient transporter interactions on BCS Class 3 drugs was explored by screening excipients that are potential inhibitors for intestinal absorptive transporters in membrane vesicles and cells. For example, by screening the FDA-approved excipients for inhibition of probe substrates of two well-known intestinal efflux transporters such as P-glycoprotein and breast cancer-resistant protein, 26 inhibitors were identified, such as dye, surfactant, and flavor agent. On the other hand, by screening FDA-approved excipients for inhibition of probe substrates of influx transporters, such as organic A9 transporting polypeptide 2B1, 27 inhibitors were identified, including dye, surfactant, antimicrobial, preservative, and flavor enhancer. Here is a preliminary assessment. Number 1, meeting the criteria for BCS Class 3 based Biowaiver does ensure bioequivalent performance in vivo. Number 2, an observation of changes in excipients is based upon the approved generic drug product formulations. Number 3, the research outcome does not mean these excipients in the stated amount can be used in all BCS Class 3 drug products. In summary, refer to the ICH M9 guidance on BCS-based Biowaiver to assess if the drug may be eligible. BCS Class 3 Biowaiver can be applied with sufficient supportive data even though the current PST does not include this option. Controlled correspondence can be submitted to request if the proposed test formulation is eligible for BCS Class 3 waiver. Last but not least, research on dissolution, modeling, and excipients continues to provide more insight to promote using BCS Class 3 Biowaiver as a surrogate for in vivo bioequivalence study, in other words, alternative bioequivalence approach in the future. I would like to thank Dr. Ping Ren, Dr. Teresa Chan, Dr. Wenchen Yang from the research team, as well as Dr. M.J. Kim, Dr. Lei Zhang, and Dr. Robert Lineberger from the management team in Office of Research and Standards. Thanks for your attention. I will be back to the Q&A session later. Thank you. Take care.
Hello, everyone. To start, I need to provide a disclaimer that this presentation represents my views and are not necessarily those of the FDA. As an overview, I'll be presenting on which drugs qualify for biowaivers and how drugs are classified according to the Biopharmaceutics Classification System, or BCS. This includes methods for permeability, dissolution, and gastrointestinal stability, along with some examples from the literature. For your reference, the information in this presentation is found in our 2021 BCS Guidance for Industry, which is available on the FDA website. BCS biowaivers are for Class 1 and 3 drugs and are described in greater detail in presentations by my colleagues in this session. Class 1 drugs have high solubility and permeability, and their products have very rapid or rapid dissolution. Class 3 drugs have high solubility and low permeability with very rapid dissolution. A drug substance is classified based on its solubility and permeability. A drug is considered to be highly soluble if its highest single therapeutic dose is totally soluble in less than or equal to 250 mL of an aqueous media. A drug is considered to be highly permeable if its absolute bioavailability is greater than or equal to 85%. Alternatively, it is considered to be highly permeable if greater than or equal to 85% of the parent drug is recovered unchanged in the urine, or as a sum of the unchanged parent drug and its oxidative phase 1 metabolites and phase 2 conjugative metabolites. Dissolution is based on both the test product and reference product. A reference product is used when comparing to an innovator drug or to a different or previous formulation of the drug. A product has very rapid dissolution if it is greater than or equal to 85% dissolved within 15 minutes or less. A product has rapid dissolution if it is greater than or equal to 85% dissolved in 30 minutes or less. The solubility of a drug substance is evaluated over a pH range of 1.2 to 6.8 at a temperature of 37 degrees Celsius. Experiments should include at least 3 different pH conditions within this range, but also include buffers that are at pH 1.2, 4.5, and 6.8. An example on the right shows the solubility results for 2 antiviral drugs using different buffers at different pHs. The shake flask method is preferred for the equilibrium solubility experiments, although other methods may be used if justified. The pH of the buffer is to be measured after adding the drug and then adjusted if needed before starting the experiment. At the end of the experiment, measure the pH again to see if there is any change over time. As for how long to conduct the experiment, you would want the solubility of that drug to reach equilibrium. The lowest measured solubility in the different buffers is then used to classify the drug substance. The experiments are to be conducted with at least 3 replicates for each of the buffer solutions used. A validated analytical method must be able to deduct degradants that is used to measure the drug in solution. A drug should be stable in the different buffers with less than 10% degradation. There are 2 types of experiments to classify a drug's intestinal permeability. The first is human pharmacokinetic studies that measure either absolute bioavailability or mass balance. The second is in vitro experiments with a standardized and validated CACO2 cell assay. Let's focus on the CACO2 cell permeability assays. Method suitability is demonstrated with model drugs that have known to be passively absorbed and have known human intestinal absorption. There are at least 5 model drugs in each of the 3 groups based on the fraction absorbed, or FA. The example on the right shows the apparent permeability, or PA/PP, of model drugs compared to their fraction absorbed. For us, labetalol was at the boundary between high and low permeability drugs. However, it is important to point out that each lab will have its own high permeability internal standard drug that is based on their results with their chosen model drugs that are used in the experiments for method suitability. Once the cell assay has demonstrated method suitability, it can be used to classify a new drug's permeability with the same protocol. First, the drug's passive absorption is established by evaluating its transport through the CACO2 cells at several different concentrations, such as 0.01, 0.1, and 1 times the highest dose strength in 250 mLs. Plus, you need to conduct a bidirectional experiment with the test drug to see if it is subject to efflux. A drug is then classified as being highly permeable when its apparent permeability is equal to or greater than that of the established high permeability internal standard. The figure on the right provides an example with fluoroquinolone drugs that were compared to the high permeability internal standard labetalol, the green bar, showing that both levofloxacin and ofloxacin would be considered to be highly permeable as their PA/PP exceeds that of labetalol. The next experiments to be considered are those for gastrointestinal stability. These are to be provided if human mass balance studies are used to demonstrate high permeability, however, with the exception of drugs that have greater than or equal to 85% recovery of unchanged parent drug in the urine. GI stability studies are also required if a drug is classified as highly permeable with the CACO2 cell experiments. The example on the right shows a GI stability experiments with venlafaxine using simulated gastric and intestinal fluids over different time periods. GI stability experiments are conducted with compendial or simulated gastric and intestinal fluids. A drug solutions are then incubated at 37 degrees for 1 or 3 hours in gastric or intestinal fluid, respectively. As with the solubility experiments, a validated analytical method should be able to detect degradants of the drug in the different solutions. If there is greater than 10% degradation of the drug, it excludes it from being classified as a high permeability drug. The last BCS method to be discussed here is dissolution with similarity based on F2 comparisons under defined experimental conditions. Experiments are conducted with the test product from a batch of at least 1/10 of the production scale or from 100,000 units, whichever is greater. The graph on the right, an example of dissolution of a new drug in different buffer solutions over a period of 45 minutes. There are a number of conditions to be considered for the dissolution experiments. A paddle or basket apparatus may be used. The volume of the dissolution media is less than or equal to 900 mL at a temperature of 37 degrees Celsius. Agitation differs between the paddle and basket at 50 and 100 RPMs respectively. At least 12 units of the reference and test product are to be evaluated, and the experiments are to be conducted in buffers at pH 1.2, 4.5, and 6.8. Additional considerations for the dissolution experiments include the lack of organic solvents or surfactants in the dissolution medium, the use of enzymes only with justification, with gelatin capsules or tablets that have gelatin coatings, the use of filtration when collecting the samples at a minimum of 3 time points, not including time 0, and the use of the same time points for both the test and reference products. To summarize, the FDA's BCS-based Biowaiver guidance provides the definition of the different BCS classes based on solubility and permeability, requirements for biowaivers of class 1 and 3 drugs, different methods used to classify drug substances and products, consideration of excipient effects, and detailed information on permeability, solubility, dissolution, and stability methods are also outlined in the guidance. And I'd like to share the references for the experimental examples that were included in this presentation. Thank you for your attention.
Good morning, everyone. Today, I'll be presenting an overview of Biowaiver aspects from a biopharmaceutics perspective, particularly our role in NDA original applications and both NDA and ANDA post-approval applications. I would like to start off with the disclaimer that the views expressed here are personal and do not represent those of the FDA. This slide emphasizes product quality. I will not go in detail as this was covered by other speakers from OPQ. As already mentioned, today's presentation will focus on biowaivers in original NDA applications. Biowaivers in ANDA original applications are handled by the Office of Generic Drugs, Office of Bioequivalence. I will also cover biowaivers in post-approval NDA and ANDA applications, followed by few case studies.
Before I proceed with biowaivers that are covered under different regulations for different scenarios, let's talk about biowaivers. From the name, we understand it is a waiver of a bio study. If the applicant intends a waiver for their product but does not submit the biowaiver request, an information request will be sent to the applicant to submit biowaiver request. A biowaiver means that the requirement of conducting in vivo bioavailability and/or bioequivalence studies can be waived per 21 CFR 320.21. Any person submitting a full NDA to the FDA shall include in the application either evidence measuring the in vivo BA or BE of the drug product that is the subject of the applications, or information to permit FDA to waiver the submission of evidence measuring in vivo bioavailability. Certain post-approval changes require support from a BA or BE study unless information to permit FDA to waiver the submission of evidence measuring in vivo BA or BE is provided. So where to find biowaiver requests in ANDA or NDA submissions? Module 1, 1.12.15 Request for Waiver of In Vivo Bioavailability Studies. Listed in this slide are the possible biowaiver scenarios under different regulations. So once the applicant submits a biowaiver request, biowaiver is granted based on one of the CFRs listed here. As you can see here, there are several different scenarios: self-evident bioavailability or bioequivalence under 21 CFR 320.22b, drug efficacy study implementation or DESI under 21 CFR 320.22c, BA or BE demonstrated in vitro in lieu of in vivo data under 21 CFR 320.22d2, IV-IVC based biowaiver under 21 CFR 320.22d3, reformulated product biowaiver under 21 CFR 320.22d4, good cause biowaiver under 21 CFR 320.22e, and BCS based biowaivers under 21 CFR 320.24 B6. The federal regulations for the biowaivers are 21 CFR 320.22, which has several subsections. FDA shall waive the requirement for the submission of evidence obtained in vivo measuring the bioavailability or demonstrating the bioequivalence of drug products. A drug product's in vivo bioavailability or bioequivalence may be considered self-evident based on other data in the application if the product meets the defined criteria. 21 CFR 320.24b. In vivo and in vitro approaches as defined are in descending order of accuracy, sensitivity, and reproducibility are acceptable for determining the bioavailability or bioequivalence of a drug product. This slide summarizes self-evident BA or BE per 21 CFR 320.22b. B1 is for injectable parenteral solution, ophthalmic or otic solutions, and should have the same active and inactive ingredients at the same concentration as a drug product that has BA or BE or PK data or is approved under an NDA or ANDA. B2 is for inhalation and it should have same API and dosage form. However, this is rare under NDA applications. B3 biowaiver is for non-parenteral solution that has the same active ingredient and dosage form and same inactive. If inactives are different, it should not affect the PA of API. Drug efficacy study implementation based biowaivers under CFR 320.22c. Drug products that were introduced to the market from 1938 to 1962. Under the Kafer-Harris Drug Control Act, requires all drug products to be effective as well as safe. DAISY program established to classify pre-1962 approved drug products as effective for at least one indication in a DAISY notice. Products with no known or suspected BE problems with a corresponding therapeutic equivalence rating in the Orange Book. These biowaivers are mostly applicable for generic applications. One of the requirements is dissolution profile similarity to reference product. The next kind of biowaiver is when the BA or BE is demonstrated in vitro. This is for drug product in same dosage form but in different strength. This different strength, availability or bioequivalence, established to classify pre-1962 approved drug products as effective for at least one for different color, flavor, or preservative that does not affect the bioavailability. The requirements include that the BA of the original product has been measured and both products meet an appropriate in vitro test. Next up, we have the BCS biowaivers based on ICH M9 Biopharmaceutics classification system. Drug products that are eligible for a BCS-based biowaiver contain a drug substance or substances that satisfy the criteria regarding solubility and permeability class 1 and 3 immediate release oral dosage forms. A drug product's BCS designation should be reviewed by the review team and the FDA BCS committee. Biowaivers are feasible during the life cycle of a drug product, both pre-approval and post-approval. This slide summarizes the different stages where biowaivers could be applicable. During formulation development, there are differences in formulation or manufacturing process between phase 2 and phase 3 and commercial formulations. In vivo bioequivalence study to the ultimate commercial formulation has to be demonstrated based on bridging, and this would be an implied biowaiver. Biowaivers are also feasible for line extensions, example new strengths or new formulations for a new patient population. Biowaivers are also possible for post-approval changes, including changes of the manufacturing formula in the manufacturing process, in excipients, in manufacturing sites, and or equipment. This slide summarizes the guidances for scale-up and post-approval changes. So far, we have discussed a number of scenarios where biowaivers could be granted during original drug approval process. Likewise, biowaivers are feasible during post-approval life cycle management of a drug product based on the SUPAC guidance for IR and MR solid oral dosage forms. SUPAC guidance provides recommendations for approval of changes related to the components and composition, manufacturing process and equipment, batch size scale up and scale down, and manufacturing site changes. And some of these changes need in vitro and or in vivo BE study documentation for approval of these changes. We have seen that a number of biowaivers require similarity testing. This slide here summarizes regulatory application of dissolution testing. Dissolution similarity testing plays an important role. It is a critical tool as it is the only product quality or in vitro attribute that relates to the rate and extent of in vivo drug release. Dissolution and similarity testing is helpful during formulation selection during drug product development and also to support CMC changes as per SUPAC guidance. Dissolution testing also supports additional strengths biowaiver. Dissolution can also act as surrogate for BE via Biowaiver request based on IVIVC or BCS class 1 or 3. Dissolution can also help in the selection of critical material attributes, critical process parameters, and critical bioavailability attributes as part of design of experiments in establishing design space and verification of design space. In addition, dissolution testing and similarity testing can help in setting of clinically relevant drug product specifications. Since dissolution is required not only as quality control but also for similarity testing for biowaivers under a number of different situations, it is ideal to develop a dissolution method with discriminating ability to critical parameters. The following information should be submitted in support of dissolution method development: solubility over physiologic pH range, selection of the equipment or apparatus, in vitro dissolution or release media, agitation or rotation, speed, media pH, assay, sink conditions, use of sinker and enzyme if applicable. Data supporting the selection of the type and amount of surfactant. The dissolution profile should be complete and cover at least 85% of drug release of the label amount or whenever a plateau, that is no increase over 3 consecutive time points, is reached. In addition, it is highly recommended to develop a method with discriminating ability. The discriminating ability could be explored with meaningful variations for CMAs and CPPs. This slide summarizes the best practices for setting of dissolution acceptance criterion or criteria. Dissolution profile data from pivotal clinical batches is desired. The acceptance criterion based on average in vitro dissolution data equivalent to USP level 2 testing, N is equal to 12. For IR products, at least 85% of the drug is dissolved, or if incomplete dissolution occurs, the plateau of the drug dissolved is reached. The specification time point should be where Q is 80% dissolution occurs. Wider specification ranges based on approved IVIVC model or PPPM will be considered. For slow dissolving drug products, 2 time point specification may be considered. For ER products, a minimum of 3 time points is recommended, covering the early, middle, and late stages of the release profile. The last time point should be where at least 80% of drug is released. If the maximum amount released is less than 80%, the last time point should be the time when the plateau of the release profile has been reached. Dissolution acceptance criteria ranges are usually around the mean target value plus or minus 10% and greater than 80% for the last specification time point. Wider specification ranges based on approved IVIVC model or PPPM will be considered. For DR products, delayed release or enteric coated, the acceptance criteria are set at the acid stage and the buffer stage as per USP. At acid stage, no individual tablet exceeds 10% dissolved at 2 hours, and for the buffered stage, Q is equal to 80% dissolution or release or the plateau of the drug dissolved is reached. The equation here summarizes the calculations for F2 similarity factor, where N is the number of time points, RT is the dissolution value of the reference pre-change batch at time T, and TT is the dissolution value of the test post-change batch at time T. Usually 12 units are recommended and 3 to 4 or more dissolution points, and time points should be the same for the test and reference. Reference batch should be most recently manufactured pre-change product. Only one measurement should be considered after 85% dissolution of both products. The percent CV at the earlier time point, example 15 minutes, is not more than 20%, and at other time points should not be more than 10%. Dissolution measurements should be made under same conditions, and the dissolution profiles should have the same time points. This case study is for a pediatric biowaiver. The fixed dose combination of higher strength is the approved strength as NDA. Waiver of in vivo BA or B studies for the pediatric strength of fixed dose combination tablet were granted per 21 CFR 320.22D2 based on same dosage form, formulation proportional similarity, and comparable in vitro dissolution profile relative to the original and current version of the reference adult strength FDC tablet. The second case study presented here is for waiver of lower strengths 5 mg and 10 mg IR tablets as a weight multiple of the 15, 20, 30 and 40 mg tablets. The 5 and 10 mg IR tablets are proportionally similar in composition to the already approved 15, 20, 30 and 40 mg tablets. Given this approach and based on previously provided data on the 15, 20, 30 and 40, the applicant concluded that in vitro dissolution and stability data would support approval of these lower dose strengths without additional clinical data. However, F2 testing showed that the profiles were dissimilar and dissolution data was not able to support or justify the approval of these strengths. The graph here shows lack of similarity between lower and higher strengths. As can be seen, the 5 and 10 milligram strengths are with the triangle and the square. They are releasing much slower than the approved higher strengths. A biowaiver was not granted based on similarity factor assessment, and the applicant was asked to conduct a bioequivalence study. Bioequivalence study showed that the 90% confidence intervals were within the required range of 80 to 125, both for Cmax and AUC. And the dissolution data, along with the additional bioequivalence study, provided support for the approval of the 5 and 10 milligram strengths. Finally, I would like to thank Kimberly Rains, my branch chief, Bhagwant Rajay, my division director, and all my colleagues in the division of biopharmaceutics for their help and support. Thank you for your attention.
Thank you so much for the very informative presentations. Now we will begin our question and answer panel. If you haven't had the chance to enter your question into the Q&A chat pod, please do so now. All right, and we'll begin with some questions for Dr. Myung. Here's the first question. If two products, which are pharmaceutical alternatives, are bioequivalent, are they considered therapeutically equivalent?
Before answering the question, I would like to justify pharmaceutical equivalence and pharmaceutical alternative first. Pharmaceutical equivalence is the drug product in identical dosage form and route of administration that contain the identical amounts of the identical active ingredients. However, pharmaceutical alternatives are the drug product that contain the identical therapeutic moiety, but not necessarily in the same amount or dosage form or the same salt or ester. If the pharmaceutical alternatives are bioequivalent, in other words, there is no significant difference in the rate and extent to which the active ingredients or active moiety in pharmaceutical alternatives become available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study, they may be considered as a therapeutically equivalent. Thank you.
Thank you so much for that response. We have another question for you, Dr. Myung. Is it necessary to show in vitro validation method?
Thank you for the question first. I think the question is whether it is necessary to perform pre-study analytical method validation for in vivo and in vitro BE studies. If I understand correctly, then the short answer to this question is yes. As I mentioned in my talk, per 21 CFR 320.2028, applicants must conduct the BE testing using the most accurate, sensitive, and reproducible approach available. Thus, the pre-study analytical method validation for in vivo and in vitro BE studies is critical and necessary to prove the accuracy, sensitivity, and reproducibility of the used analytical method. Thank you so much.
Thank you for sharing that response. Our next questions will be for Dr. Rong Wang. In a situation where FDA dissolution medium, which specifies 75 RPM in 1000 ml, has been used, but ICH M9 specifies paddle apparatus of 50 RPM, basket apparatus to 100 RPM, with volume of dissolution medium 900 milliliters or less, which one should be adopted?
Thank you for the question. First, I want to clarify that the dissolution tests recommended in ICH M9 guidance are for generic drug products with BCS waiver requests. If the applicant intends to submit an ANDA with a BCS waiver request for its generic drug product, among other recommended information, the applicant should conduct the multi-pH dissolution tests following the recommendations elaborated in the ICH M9 guidance. If the applicant is adopting the FDA recommended dissolution method for quality control and stability purpose, the applicant should also submit the dissolution testing data using the FDA recommended method. Whether the FDA recommended method is suitable for the proposed generic drug product for quality control purpose will be determined by the Office of Biopharmaceutics within OPQ. Thank you.
Thank you so much for that response. Now we have one more question for you, Dr. Wang. Can we perform PBPK study in place of clinical endpoint studies?
Thank you. If the applicant proposes a PBPK study instead of the recommended clinical endpoint study for establishing BE for its generic drug product, the applicant is recommended to request a meeting prior to the ANDA submission, first with its justification and the relevant and appropriate data to discuss with agency on why their proposed PK study can be considered as a sensitive, accurate, and a reproducible method for establishing BE for its generic drug product. The applicant could also use the meeting opportunities to further clarify regulatory expectations regarding their generic drug development program. Thank you.
All right, thank you for that response as well. Our next questions will be for Commander Yi Zhang. So the question is, for a BCS3 based biowaiver, please clarify the reference product to be used for comparison with regard to formulation similarity and very rapid dissolution, RLD or RS.
Thanks, Nora. So for ROD, which is the reference list product, so usually it is a new drug product held by innovator. So RS is the reference standard product, which is designated in the current Orange Book. So for RS, it could be either ROD or generic.
So, for example, in some cases, the generic drug becomes RS when the ROD was discontinued, but not due to safety or efficacy issues. In another cases, or could be the ROD had been discontinued for a certain period of time, but then come back to the market later. So in short, you always should refer to the current Orange Book for the reference standard product, which can be used for your BCS3 based Biowaiver request. Over. Thank you.
Thank you for sharing that information. All right, we have one more question for you. Can literature data or alternative scientific justification for solubility be used as pivotal data to qualify a drug substance for a BCS3 based biowaiver?
Thanks again, Nora. So the answer is no. You should always conduct your own solubility study to establish the high solubility of the requested drug substance, which are for waiver, and submit it as pivotal data. However, the literature data may be submitted as a supportive. And in the meanwhile, with regard to the permeability data, you may refer to either NDA or any publicly available information for BCS3 based Biowaiver. Over. Thank you.
Thank you so much. All right, now we'll move on to some questions that we have for Dr. Donna Volpe. Can animal studies be used for permeability classification?
Thank you for the question. At this time, the BCS guidance limits permeability determinations with human clinical studies, whether they be for absolute bioavailability or for mass balance, or if you want to do an in vitro study, those can be conducted in the CACO2 cell line permeability assay. Thank you.
Great, thank you for that response. I guess a related question to that, are CACO2 cells the only cell line to use for permeability experiments?
Thank you for the question. And yes, at this time, only CACO2 cells are being, results in CACO2 cells are being accepted for BCS classification for high permeability. I do know that there are other cell lines available that folks will use in their labs for permeability experiments, but only the CACO2 cell lines are being accepted right now. Thank you for your question.
And thank you so much for that clarification. Now we'll move on to some questions that we have here for Dr. Haritha Mandula. First question, what are the alternatives if the conditions under a biowaiver regulation are not met?
So it depends on actually what kind of regulation is being sought. So if the biowaiver is not met, there are alternatives like, can it be adequately bridged based on other justification? Does the changes not affect the disposition of the drug? And then there are also modeling strategies, to demonstrate that the changes being made do not affect the absorption or even the disposition. And in addition to that, there are always, the ultimate option would be the conduct of a BE study. But even before going there, it should be evaluated, what effect it has and why is it not being met. Like example, if it were the case of F2 similarity not being met, it has to be evaluated why it is not met. Thank you.
All right, thank you for that information. Follow-up question to that, if F2 similarity is not met, what are the alternate options?
So as I mentioned, it has to be seen on why F2 similarity is not met. Sometimes, is the noise coming from like the experiment or is something else going on? Is there possible strength-dependent dissolution? That could be one possibility. Or is there compositional proportionality? In the absence of that, sometimes the options could be like using a bracketing kind of approach, like having a B study on the highest and the lowest strength, and the in-between strengths would be eligible for a bio-waiver. And as I previously said, if the conditions are not met, I mean, of course, in vivo studies would trump, all these situations. Is there dose proportionality studies conducted in vivo? So, some of the scenarios could be in this regard. Thank you.
Thank you for that response. We actually have another question that came in for Dr. Rongwang. For ANDAs, do we have to demonstrate permeability using different studies, or will information on bioavailability from labeling suffice?
Thank you for the question. So if the ROD labeling explicitly indicates the absolute bioavailability is greater than or equal to 85% based on the in vivo bioavailability study or mass balance data, the applicant could reference the ROD labeling information to support the high permeability of the drug substance. Otherwise, the applicant should submit its own permeability study data to demonstrate that the drug substance is highly permeable. Thank you.
Thank you again for that information. We'll continue with another question for you, Dr. Wang. Can an applicant submit an ANDA for a generic drug that is not the same as its RLD?
Thank you for the question. The short answer is yes. An applicant may submit an ANDA for generic drug that is not the same as its RLD in terms of active ingredient in a fixed combination drug product or a different route of administration, dosage form, or strength, then the applicant first must obtain permission from FDA through the citizen petition process. So this is called a suitability petition. FDA will not approve a suitability petition if investigations must be conducted to show the safety and effectiveness of the proposed generic drug product with difference from its ROD. And a generic drug that is approved under suitability petition is not considered therapeutically equivalent to its ROD because therapeutic equivalence must be pharmaceutical equivalence, while a generic drug product approved under a petitioned ANDA is not pharmaceutical equivalent to its ROD due to the differences I just mentioned. Thank you.
Thank you again for that response. We have another question that maybe for you or Dr. Myung as well. Is there any difference for studies and data submitted for 505J versus 505B2?
Thank you for the question. I think I can answer this question first, and I'll ask Dr. Myung to add later. So the short answer is yes. There may be differences in the types of studies, data, and information that may be necessary to support approval of drug products proposed in ANDAs compared to 505(b)(2) applications. 505(b)(2) applicants have significant flexibility in the types of studies, data, and information they may submit in a 505(b)(2) NDA to support the requirements for NDA approval. The types of studies that may be submitted in 505(b)(2) NDA could include clinical investigations to establish the safety and or effectiveness of a drug product. If a clinical study, meaning any experiment other than an in vivo bioavailability study is necessary to demonstrate the safety or effectiveness of a proposed drug product. Generally, this type of study goes beyond the scope of information that is required under 505J, for the approval in an ANDA. The applicant is recommended to contact the OGD first for guidance prior to submission of an application when data outside the scope of the ANDA pathway is considered necessary for its application. Dr. Mian, do you want to add, have anything to add?
Thank you, Dr. Wang. I think Dr. Wang covers mostly. I would just like to add some fundamental comparison between two pathways. Both 505B2 and 505J applications partially relies on certain data from an already approved drugs called as a reference listed drug. I think we heard multiple times, RLD. However, as Dr. Wang mentioned, in the 505B2 application, there are intentional differences between the proposed drug and the RLD, such as differences in active ingredients, dosage form, route of administration, strength, indication or other conditions of use, bioequivalence, or bioavailability. However, under application under 505J pathway, do not need to submit the clinical investigation to establish the safety and effectiveness, since the proposed test product will have the same active ingredients, same dosage form, same route of administration, same strength, same indication or other conditions of use, and the same bioequivalence. Thank you. Thank you so much, Donna.
All right, Dr. Myung, while we have you on the line, we might have one more question for you. If a product-specific guidance is different from the general guidance, which guidance should be followed?
Thank you for the question. That's a great question. If a product-specific guidance or PSG is available on FDA's website, the recommendations in the PSG generally supersede those described in the general guidance. Thank you. Thank you for the question.
Okay, thank you so much for that response. Looks like that might be all the time we have for questions. I want to thank all our presenters again for all the excellent information they've shared today, and also for taking the time to respond to questions in our Q&A sessions here. I'm now going to turn it over to Jun Mukike for some closing remarks.
And it looks like we might be having a little bit of technical difficulty with Jude's microphone. Jude, can you check to make sure that the microphone you're using isn't double muted? We have you unmuted in Adobe Connect, but we're still not hearing you. So can you check on your microphone, please? All righty, it looks like we're still having some trouble. So Hale, if you do want to jump in here, you'll need to connect your microphone, and then you can jump in as needed.
Would you like me to cover?
Yes, please go ahead and
Okay,
jump right in.
Thank you. I apologize. I think Jude is having a little bit of technical issues. I'm Sohail Mossadegh. I'm Director for USB's U.S. Regulatory Affairs. Special thanks to Captain Stoddard and Lieutenant Lim for leading such an efficient and really well-coordinated event. I have a much better appreciation of what's going on in the background and the planning for these events, and it is an amazing amount of work. I'd like to thank, on behalf of PQM Plus and USP and Jude, all of the speakers and participants from around the world for joining us during this 3-day conference. It's been a very exciting collaboration amongst the US FDA, USAID, WHO, and USP colleagues. As Dr. Woodcock said, the overarching goal of this meeting has been to share knowledge and resources that can be applied to strengthen the global pharmaceutical supply chain and really help ensure access to these important medicines, including but not limited to TB medicines. We are grateful to the FDA for their support in developing this conference and making it possible for everyone for USAID, USP, and PQM Plus to reach a much wider audience. I'd like to highlight the many technical areas covered by the experts, speakers, especially important for those that may have missed some of the events. The recordings are available afterwards, and I find these SBIA recordings to be immensely useful. The topics covered were the WHO's drug pre-qualification process and the collaborative procedure for accelerated registration of WHO pre-qualified medicines. Specifically how LMIC can benefit from these procedures and ensure timely access to quality assured medicines. Opportunities for international engagement, regulatory coordination, convergence, and harmonization. A topic I found very interesting was the new drug application as well as the abbreviated new drug application. Great overview. Bringing new TB drugs to the market and looking at regulatory pathways and designations utilized by the FDA to facilitate and expedite drug development for serious or life-threatening conditions. Another topic was the drug development programs for TB, including clinical trial design, study population, and endpoints, including surrogate endpoints. Also covered were the tentative approval pathways for drug products to quickly get critical antiretroviral medicines found acceptable for use in areas of greatest need. Expanded access, also known as compassionate use, as a potential pathway for gaining access to investigational medicinal products. And today, the nitrosamine compounds topic by Dr. Raw and Murnau, which are potent genotoxic agents and are referred to as cohort of concern compounds, and the conditions that may introduce nitrosamine impurities to pharmaceutical active ingredients and drug products. In the final, but not to be forgotten, bioequivalence for generic drug products, including various types of BE studies that may be submitted in support of NDAs. Overall, our hope is that you will take a deeper understanding of regulatory best practices and resources available by FDA and ways to apply them to meet your individual national health needs and goals, specifically for strengthening the reliance mechanism for ensuring timely access to quality assured medicines. With that, thank you again. I wish everyone a good day, and thank you, everybody.